So the first test that most kids would have when we think they might have an underlying genetic condition is called a microarray. And a microarray looks for bits of missing or extra genetic material. So the test covers all of the child's DNA, looking for whether on each chromosome, on each bit of genetic material, is the right amount of genetic material present, or is there too little or too much?
An easy way I think of a chromosome, even though it's not scientifically accurate, is to think of it like a piece of string, and the genes are like beads beaded onto the string. So it's a way of organising our genes in a neat way in our cells. And a microarray is looking at all of the chromosomes and looking for small missing pieces or small additional pieces. But the problem with the microarray is it's not sensitive enough to pick up very small changes. So in order to do that, you need to know specifically which gene you want to look at, and you need to read the entire sequence.
Exome sequencing is a new type of genetic test where all the coding regions of all of an individual's genes are spelled out. So in our body, we have thousands of genes, which are the instructions to make our body work for us to be healthy and develop normally. And there are about 20,000 genes altogether. And the most critical part of a gene is what's called the coding region of the gene. That's where the instruction to make a protein, that's where it resides in the gene. And that's where most of the mistakes lie if that cause genetic conditions. So an exome is sequencing all those important coding regions of all of the genes all at the same time, all 20,000 all at once.
If we think of the genome, which most of us have heard of a genome, the difference between a genome and an exome is the exome is the important part of the genome. So there's a lot of junk in the genome itself. So it's sort of like thinking of a book, and the start of it, the cover and the introduction are not so important. It's the meat of the book that's important. And the exome is like the meat of the book, excluding all of the other parts that don't really matter.
So what we're doing when we're analysing a person's genome or all of their DNA is we actually get the DNA from multiple cells, and then we actually chop up that DNA into little pieces, and we do what we call sequencing those little pieces. So when I say sequencing, what that means is that we're actually looking at the letters that code for the different parts of that DNA in a sequence. So we actually sequence those little fragments, and those fragments of DNA, then we actually put them back together like a big puzzle. And that, once we've put that puzzle together, that allows us to see where the differences might be in a person's DNA compared to what we know is the normal DNA in the general population.
Another easy way I try and imagine a gene change is with our genes really are like a book of instructions. So that book lists all of the ingredients that the body needs to put together to create a protein. And what the genetic testing is doing is reading that entire book and looking for words that are incorrect or ingredients that are incorrect. Now, if you're following a recipe and you change a spice, so let's say you change from cinnamon to nutmeg, it's not going to make much difference to the end result. But if you change something like from eggs, you take eggs out and you put extra flour in, you're going to significantly change the end result. So what the laboratories need to do once they discover a change is figure out whether it's a fairly harmless change, like changing a spice, or whether it's a significant change. And that's a very labour-intensive process.
Exome sequencing has brought with it the power to get a diagnosis in a third to a half of the children where we suspect a genetic condition. But what that means is there's still a half to two-thirds who are not getting a diagnosis from that technology. That's going to improve over time, both with us getting better at using exome sequencing, the sequencing technology improving, and also we believe that whole genome sequencing will eventually become the test of choice in children with undiagnosed syndromes.
So the other really key things that are improving, will improve things over the next few years, are the technology for the sequencing. At the moment, the sequencing technology can only sequence fragments of a certain length, but that really limits how accurately we can detect the variants in a person's genome. as we lengthen the fragments that we can sequence, so from 100 base pairs or 100 letters long up to thousands of letters long, then we can very accurately place that DNA in the context of the reference genome and detect variants that we weren't able to detect before. Some of those are like triplicates, so letters that are repeated in triplicates. Those kind of variations we're unable to detect at the moment. Those variations we will get better at detecting. Along with that technology, with the long reads, we'll also get better at detecting parts where the DNA has been lost, or what we call deletions, and parts where the DNA might have been gained, or what we call amplifications or gains.
It's really important for families to know that getting a negative exome or not finding the diagnosis is not the end of the journey. We know that when we continue to analyse that exome data over time, new genes are discovered, new conditions are discovered, and we get another 10% of our diagnoses from that analysis down the track.
At the moment in Victoria, we're seeing new information about genes being related to certain conditions happening in a sort of a 6 to 12 month time period. So I would anticipate that somewhere between 6 to 12 months might be a suitable time for checking whether there's any new information related to your condition. And another time point would be to wait if a new technology seems to have been implemented, then potentially your condition might benefit from that new technology.